Okay, so the last nutrient that we're going to spend a lot of time on is lipid, or are the lipids. Now, like I have mentioned in several other videos, the first thing that has to happen for lipids to truly be digested and then, of course, after that be absorbed, is that they first must be emulsified. Emulsification is a physical or mechanical digestive process, okay? And so what I want to do is I want to focus you on this little breakdown thing of what emulsification really is. And let's walk through that and talk about it. And then we can get into the enzymes that do the actual chemical digestion. So let's read here. It says fats are insoluble in water, so they need special pre-treatment with bile salts to be digested and absorbed in the watery environment of the small intestine. Okay, so basically what that means is fats don't like water, right? They don't dissolve in water. Fats like to be by other fats. So if you can see this, I know it's really light kind of pink color here. If you can see this right here, this is a fat globule. And these little green lines are bile salts. And so what bile salts do is they insert into the fat. And bile salts have two kinds of chemical sides to them. One that likes fats and one that repels fats and likes water. And so when you put bile salts into fat, the part that likes fat will stay inside and the part that repels fat will stay on the outside and start separating the fats from each other. That's exactly what bile salts do here as you're seeing this. It's actually what detergents do too, to your oils and your dirt and stuff on your clothes and on your dishes. Okay, so it says bile acids play their first critical role in lipid assimilation by promoting emulsification. As derivatives of cholesterol, bile acids have both hydrophilic and hydrophobic domains, which means that they're amphipathic. That means they have two different sides to them, like I said. One likes water, one doesn't. Okay. On exposure to large aggregate of the triglycerides, so lots, what that means is just a big chunk of fat that likes, fat likes fat, sticks to each other. The hydrophobic portions, the water-fearing parts of the bile acids, intercalate into the lipids. So they go into the lipids. The hydrophilic parts stay on the outside. Okay, and so it just starts more and more bile acids and bile salts coming in. We'll just continue separating the fats from each other. Okay, so I've written a little bit on the bottom here. It says emulsification does not break chemical bonds. It just reduces the attraction of fat droplets to one another. So it just physically separates fat. Okay, so let's go back up here to the top. In order for us to digest lipids, we've got to go from triglycerides that are clumped together, unemulsified triglycerides. Then we need to emulsify the triglycerides. And then we need to break them down into glycerols and fatty acids. Reminder, we wrote this earlier, right? Fats, triglycerides, down to 3 fatty acids and a glycerol, or a monoglyceride. Okay, so over here, let me move. Okay, it says mouth. We know that there are enzymes there, but not a whole lot of digestion's happening because you need emulsification. You got to separate the fats from each other to actually get the enzymes in to break apart the fats. Okay. Stomach, also an enzyme, but not much digestion's really happening here. This is where it's all going to happen, down here at the small intestine. We get the bile salts from the liver. Remember, the liver produces the bile salts. Gallbladder stores and concentrates bile. Okay, that's for emulsification purposes. Then the pancreatic lipases are really the enzymes that do the work here. Notice that there are no brush border enzymes for lipids. Okay, so pancreatic lipases coming from the pancreas are going to break apart the triglycerides. Now here's where it gets a little bit weird. It's the lipid absorption. Lipid absorption is super different than what we see for carbohydrate and protein absorption. I'm actually going to skip a slide and then I'm going to come back to this one because this one's a little bit more simple. So let me move over here. Okay, so it says lipid absorption. So here, what you're seeing in this picture is this yellow thing is a villus cell. So this is our small intestine cell. Inside the small intestine cell in the villus, remember we have the capillary and lacteal. You know, I'm going to try and draw this. I'm not the greatest drawer. You guys know that. But here on my drawing, okay, here's the villus. Inside we have the capillaries, right? So we've got capillaries. Okay, and then we also have another vessel. This is the lacteal. Okay, what we're seeing here in this image would be like, these are the cells. This is the yellow thing in the image. Okay, so that's the villus cell. Here's the capillary. There's the lacteal. So when we look at our image on the slide, we're seeing here's the yellow cell, here's the capillary, and here's the lacteal. Okay? So on this side over here, this is where the food is. This is the inside of the small intestine, the lumen of the small intestine. And so what we've got here is we've got our fatty acids and glycerol. So the pancreatic lipase has come in, it's broken apart the lipids, and now we're down to its component parts, fatty acids and a glycerol. Okay, so the triglycerides have been broken down. Those things, when they're broken down like this, the triglyceride components, when they're broken down and we have bile salts wrapped around them, we call that a micelle. Okay, write this down so we're good to go on this. A micelle are the fatty acids and the glycerol, or the monoglyceride. By the way, these are the components of a triglyceride. Okay. So it's these guys surrounded by bile salts. Okay. All right. So this is what happens. This micelle forms inside the small intestine lumen, and it moves towards the villus cell here. The components of the fat are going to move into the villus cell via simple diffusion. Why can they do that? Why do the other ones have to be actively transported, but this one can be moved in via simple diffusion? I hope you're thinking this can happen via simple diffusion because the plasma membrane is made of phospholipids, right? And these are lipid components, so they can move right through the plasma membrane via simple diffusion. But look what happens right when we get into the cell. It's hard to believe this after all of that work and all of that chemical digestion, but the triglyceride reforms inside the villus cell. 3 fatty acids and glycerol come back together inside the villus cell. Okay, so we have this is occurring simply, right? This chemical digestion to break us down to fatty acids and glycerol is simply there to get the fats to move into the small intestine wall cells, the villus cells. These are simple columnar epithelial cells. So once we have the triglyceride recombined here, and we get a protein coat on top of that, okay, protein coat. Notice, this is something new here. Proteins inside the villus cell wrap around that triglyceride, and we create something called a chylomicron. You might say, why do we make this protein coat? What is the point of this protein coat? The protein coat is there to allow that triglyceride to be transported through the body fluids. Remember, lipids don't like fluids, and so we have to coat these lipids with proteins, otherwise they won't transport through the body fluids. Okay, so a micelle forms in the lumen of the small intestine. Okay, a chylomicron is a triglyceride recombined that's coated with proteins. Okay, so it's a triglyceride and coated with proteins. This forms in the villus epithelial cell, which is a simple columnar epithelial cell. Okay, and the chylomicron, we call this a lipoprotein. Look at the terms here. lipid plus protein. Okay. The whole point of this is this allows for lipid transport. This is important for lipid transport. Okay. Again, we're going to talk more about chylomicrons in the next chapter. Okay. So, excuse me. We have this protein coat, and take a look at how we get this chylomicron out of the villus epithelial cell. We actually use exocytosis here. So we use vesicular transport, and we get this whole big structure out via exocytosis. Notice that the chylomicron does not go into the capillary. It goes into the lacteal. Remember when we talked about lymph vessels, we said that lacteals are much more permeable, right? So the lacteal is going to have those mini flaps that we have talked about before, and those chylomicrons can diffuse into the lacteal because they have much more permeable walls. All right, so this one has a lot more writing. Oh, I don't need more to go. We'll do it over here first. Lipid absorption steps. Here's our fat globule. Here's our emulsification, right? Bile salts. Okay. We get the pancreatic lipase to come in, and we create these micelles, which are going to have fatty acids, monoglycerides, and the bile salts wrapped around them. They move to the villus epithelial wall. The bile salts stay in the small intestine. The fatty acids and monoglycerides move into the villus cell here. They recombine to form triglycerides, and then chylomicrons form inside the epithelial cell. The chylomicrons leave via exocytosis, what we're seeing here, and they move into the lacteal. And so they are carried through the lymphatic system. They will move through the lymphatic system until the lymph returns to the blood, way up at the subclavian veins, right? All right. So here I've written the lipid absorption steps one by one. We get micelle formation. The fatty acids and monoglycerides will leave the micelle and diffuse into the epithelial cells of the small intestine. Then they recombine to form triglycerides, and then we have protein-coated lipids called chylomicrons that are formed in the villus. We use exocytosis. The chylomicrons leave the epithelial cells of the small intestine, and they enter the lacteals. And then the lymph transports the fats to the bloodstream. Again, you can look at this image as a simpler form of that. As far as nucleic acid digestion goes, we're going to go from nucleic acids, pentose sugars, nitrogen-containing bases, to phosphate ions. So all the components of the nucleic acids. We're talking about DNA and RNA. All nucleic acid digestion occurs in the pancreas and in the small intestine with the help of pancreatic enzymes and brush border enzymes. Nucleic acid absorption is very similar to protein and carbohydrate absorption. I will not test you on nucleic acid digestion or absorption. I'm just giving you that information in case you need it for the future. As far as water absorption goes, most of it is going to occur in the small intestine. About 95% occurs in the small intestine by osmosis. The rest is absorbed in the large intestine. We need a little bit of water to soften the feces. The longer that feces are in the large intestine, the longer that water absorption can take place and the harder the feces can become. This image, like I said at the beginning of one of the other videos, is an excellent source to talk about chemical digestion here, where things happen, and absorption and how this occurs. I highly recommend this as a source for you to study. The remaining 3 slides are just FYI information about various digestive situations like gluten sensitivity, celiac disease, lactose intolerance, and baby allergy to foods. You certainly are welcome to look at this and read it. I would be happy to answer any questions about this, but that is not going to be tested on.